So in the last lecture, we started talking about mitosis. And mitosis began with prophase, where the nuclear membrane, this is the nuclear membrane right over here in blue. Well, it's actually really in white. But notice that this nuclear membrane, which is in white, but I'm drawing it in blue, it starts to disintegrate. And these are the fragments of the nuclear membrane or the envelope after prophase. So notice in metaphase, we don't have any of the nuclear envelope fragments anymore. Another thing that occurred in prophase was that these centrioles form microtubules. And these microtubules bind on to the centromere of the duplicated chromosomes. In metaphase, the chromosomes align along the equator of the cell. In anaphase, so we stopped at anaphase, now we're going to talk about anaphase. So we're still in mitosis, we're still in anaphase, the anaphase part of mitosis. So the sister chromatids which are the duplicated chromosomes here. The reason they call them sister chromatids is because when the DNA is replicated, they make identical chromatids which look alike. That's why they call them sister chromatids. So these sister chromatids split, and we call this process disjunction. Disjunction. And then we move into telophase. In telophase, there's a couple things that occur in telophase. So telophase, I'd like to think of as the opposite of prophase. So what occurred in prophase? We saw that in prophase, the nucleolus disappeared, the nuclear membrane disappeared, the chromatins condense into chromosomes, and these centrioles right over here form these microtubule spindles. So what do you think will happen in telophase? Well, the nuclear membrane reforms. Chromosomes decondense now. So we don't have to package these chromosomes nicely because we're no longer, they no longer need to divide at the moment. So the chromosomes decondense back to the chromatins, the uncondensed form of chromosomes. Decondense back into chromatins. And then there's also a process called cytokinesis. And cytokinesis, so I've looked at a couple of articles, and some articles say that cytokinesis occurs in telophase. Other articles say that cytokinesis begins at the end of anaphase and expands throughout telophase and still continues after telophase. So there's a little overlap on when cytokinesis actually occurs. But for simplicity, I'd like to just group cytokinesis occurring during interphase. So in cytokinesis, the cytoplasm actually splits up too. And as a result, we have two identical cells. So the whole objective here was that we had a skin cell and we made two skin cells. Oops. And this whole process occurred via the cell cycle. First there was G1, then there was, and in G1, remember certain cells can actually go into, so think about the cell cycle like this. First there's G1, then there's synthesis, then there's G2, and then there's mitosis. Something I wanted to mention about G1. Some cells hang out at an offshoot called G0, where they just don't continue to divide because they're so specialized. And those cells are nerve cells and muscle cells. There are certain cells that hang out in G0 sometimes, and they hop back into G1 and actually do divide when stimulated. And those cells are liver cells. So sometimes alcoholics who actually abuse their liver, part of their liver actually gets destroyed, and when those liver cells are destroyed, some of those liver cells go from G0, when stimulated, back into G1 and divide. So that's pretty cool, I think. So that's all for mitosis.